fine. So all I'm going to do is I am literally going to go through a whole topic tonight. I'm going to go through cell biology. Okay, now the last time you did cell biology was with me in year 9, so that was a very long time ago. Okay, and just to remind you, your mock is going to be on all the year 10 material. So yeah, I'm going to do kind of like a mind map. As I do a mind map, I might ask you some questions, okay? Or if you've got a question, you ask me. I'm planning for this to probably 4:15, probably 4:30 at the latest, if that's okay. Or do you want it to go longer? I don't know. No, okay, right. So, okay, so cell biology. It's we'll go for the first thing. We'll go towards the structures first. So we'll talk about the structures and function, okay, of different cells, first of all. So what kind of cells have we got to talk about? Plant and animal cells, yes. So we'll do animal first. Okay, so very, very simply, an animal cell, you've got very simplistic this, but we'll go anyway. So you've got a nucleus. What does the nucleus do, please? Yeah.
Doesn't it have DNA?
Yeah, it contains DNA. Well done. So put that actually. So it contains DNA. Good. And it actually controls the cell. Can anyone tell me the thing you shouldn't say?
Is it that you shouldn't say it's the powerhouse of the cell?
It's not the powerhouse of the cell anyway, but some people call it the brain of the cell. It's not a brain, okay? All right, so never use that term. I don't know why I'm mentioning it, because some of you are mentioning it now. Anyway, so you've got the nucleus. You then have other features. The interior thing is called the cytoplasm, where, well, what happens in the cytoplasm? Anyone remember, Amy?
The reactions.
Yeah, chemical reactions, well done. So chemical reactions, very good. And then we've got these structures here that are involved in respiration. So what's involved, what is the site of respiration? Yeah,
Is it mitochondria?
it is mitochondria, well done. So mitochondria. Some people call it the powerhouse of the cell when it's not, it's where respiration occurs. So respiration. Then we've got dotty things. We're going to call them ribosomes. And ribosomes is the site of protein synthesis. And the last one is the cell membrane. So you've got the cell membrane. What does the cell membrane do? Yeah.
Controls what goes in.
Yeah, so it controls what goes in and out, basically. Goes in and out. Good. Cool. So there you go. There's animal cell. That's the animal. Then we've got plant cell. I'm not going to go through all of the parts. I'm just going to go through the specific parts we need to know. So plant cells. So they got 3 differences Right, can anyone name me one difference between a plant and animal cell? Katie? Good. So the first difference is that one is the cell wall. It's made of cellulose, cell wall. It basically strengthens the cell. Yeah. It does have chloroplasts. Yeah. You know what happens in a chloroplast?
Is that where the sunlight is absorbed?
Yeah, for photosynthesis. That will be the next topic if you haven't started it already. I think you're doing photosynthesis next. No, you're not. You're not in year 10. You've done photosynthesis, what we're talking about. Right. And last one. Yeah.
Is it the permanent vacuole?
Well done. Very good. Permanent vacuole. And that contains cell sap. And that keeps the cell turgid. Good. Turgid just means firm. There you go. So obviously it contains all the same things as an animal cell, but those are the three differences. So they do like to ask you that about the differences and similarities. Dead simple. Right, whilst we're on the cells, we're going to do the next one, which I'm going to do is prokaryotic. Prokaryotes. Anyone give an example of a prokaryotic cell? Yeah.
Isn't it like a plant or animal cell?
No, it isn't. It's very different. So collectively, plant and animal cells are known as what we call eukaryotes. All right. Whereas prokaryotes are things like bacteria. So eukaryotes are plant and animal cells. Whereas prokaryotes are basically things like bacteria. So you need to know the following. So here's a bacteria. Now, AQA, you know, sometimes when you've seen them, they've got tails on them, called flagella. Well, in AQA, they don't really talk about flagella. So we're not talking about flagella today, which is a shame. I like flagella. It does have a different interior. So the interior, it's got loose bits of DNA. Okay. It's got ribosomes, smaller ribosomes. So let's label that. So we've got, is it linear DNA? No, we're going to call it loop of DNA. So loop of DNA, so that it doesn't have a nucleus, so no nucleus. And then it has similar things. It has like a cell wall. It does have a cell wall, thicker cell wall though. It's made of different things. It also has little round DNA called plasmids. So we do say, here's a little good, I like this statement. So the DNA is free in the cytoplasm. So DNA is free in cytoplasm. So the big difference is that basically it doesn't have a nucleus. whereas a eukaryotic cell does. That's that one. I'll just give you a moment just to catch up. I'm going quite quick. Right, very quickly, I'm not going to go through every single specialised cell, okay, but we need to talk about specialised cells. Right, so what is a specialised cell? How would you describe it? Yeah?
Is it a cell that has like a specific job?
Yeah, so it's got a specific function, okay? So specific function. And it's also what we call differentiated. So differentiated. And basically animals. So animal cells, you need to know about the following. You need to know about sperm. You need to know about muscle cells. You also need to know about variety. Nerve cells, you need to know. Red blood cells. And then plant cells, you just need to know about a couple that you've done already. So root hair cells. Phloem and xylem. Talk about those in a minute. Phloem, xylem, and pulsate cells. There you go. So you might be asked about what their function is, their adaptation. So for example, I mean, sperm cells, what do? They fertilize an egg. Can anyone tell me some kind of adaptation that they have? Yeah? They've got a tail to swim, very good. Okay, what do they have in the sperm, in the head of the sperm? Yeah? Yeah, they've got mitochondria as well, got lots of mitochondria, and they've got enzymes in the nucleus. So that's the kind of thing. So tail, I'll just keep it brief. Mitochondria. Muscle cells, again, lots of mitochondria because they need to release energy. Nerve cells, they're very long. Okay, they're very thin. They've got branched endings. They've got what we call myelin sheaths to insulate them. Okay, so I'm going to put that actually. So they're insulated for electrical impulses. What's the key feature about red blood cells? Well, red blood cells, they carry oxygen. What do they not have? You remember, what don't red blood cells have that all the others do? Remember, yeah. Yeah, so no nucleus. Well done. No nucleus. So stuff like that. Usually, in the exam, they would give you some information. Okay, but those are the main ones you need to know about. Root hair cells. So you've done a lot about root hair cells. So what do they do? Do you know what they do? So they absorb water and mineral ions. They're adapted because they got a large surface area. Again, they contain mitochondria. Whenever you see mitochondria, I want you to think about active transport. Okay, so it means there's lots of energy to move things against the concentration gradient. Okay. Anyone remember what the phloem transports? Yeah?
Is it like dissolved sugar?
Yes, dissolved sugars. Well done. So dissolved sugars. The phloem cells are alive. Okay, but dissolved sugars. Well done. The xylem transports water and mineral ions up the plant. So here's a little extra then. I know this is related to photosynthesis. Yeah, bioenergetics. No, it isn't. Organisation. So yeah, so phloem, do you know what the movement of sugar, what process is it called? Can you remember? Yeah. Yeah, well done. It's translocation. Fantastic. Translocation. And the other one for where water and mineral ions taken up the plant. Yeah. Transpiration, well done. And you do need to know about those for organisation. So there you go, it's a crossover. Can I say as well, when you get an exam question, sometimes exam questions don't just have one thing in them. So if they might start off with you labelling a plant cell or something, and then explaining what it does, and then linking it to another part of the spec. Okay, so you won't just get a question on cells, you might get a question on cells that turns out to be something about transpiration, for example. So they can ask you multiple things. Sorry, that's the drain. Does that all the time. Palisade cells, again, they're found, found in the leaf. What are they specialized to do? What do they have lots of? Can you remember? Yeah? Yeah. Well done. It's got lots of chloroplasts, actually, photosynthesis. Okay, they're very tightly packed at the top of the leaf. Top of the leaf to get the most sun. There you go. Good. So that's specialized cells. Dead easy. Right, so how do we study cells? What do we use? Yeah, a microscope, yeah. So let's talk about microscopes. This is quite quick, this section. Oh, look at that. I just haven't planned it very well, have I? There you go, microscopes. Okay, so they'll ask you about two types of microscope. They might ask you about light microscopes and electron microscopes. So we just need to know some differences between the two. So I'm going to do light up here and electron. Okay. So light microscopes, they are relatively cheap. So they're quite cheap. Whereas electron is expensive. Light have low magnification, whereas electron is high magnification. Light is also low resolution. We'll talk about resolution in a second. High resolution. So I think we know magnification is how many more times bigger something looks. Does anyone remember what resolution means? It's quite a hard term. Yeah. Go on, Serena.
No, I don't think it's right.
Okay, just have a go. It doesn't
It's
matter.
about how clear something is, or-
Yeah, it's about clarity. So it's basically how close two objects can be together before they become blurred. Yeah. Yeah, so it's related to pixels as well, but in your exam you need to know resolution is about basically so how close two objects can be seen together. Right, so that's resolution. Right, the thing about light is you can see living samples. So living samples can be seen, whereas electron, unfortunately, they are dead samples. Can anyone tell me why they are dead samples in the electron microscope? Yeah. No, it's not to do with that, because there's no light. It's electrons rather than light. Yeah.
Does the electron one have to be in the vacuum?
Yeah, it has to be in a vacuum. Well done. It's in a vacuum. So there's no air. So that's why they're dead. So I should have started this one off actually. So you're using light for the image, using light for an image, whereas electrons, you're using electrons. So electrons have a smaller wavelength. So they've got, if you remember wavelength, you've got a really small wavelength, whereas light has a longer wavelength. Ooh, that's terrible. Physics won't like me. There you go. Right, they will ask you, well, I don't know whether they will in this exam, but they might ask you about images. Okay, so let's talk about images. Does anyone remember the way I taught you to remember how to work out magnification or actual size or image size? Anyone remember? Yeah? I am. Well done. Well remembered. Fantastic. So yeah, we did the I am triangle. I am. Okay. So I am. So we can work out if it's the image. So image equals, if we cover it up, it's actual times mag. The actual size, which it usually is, it's usually the actual size, is, if we put our finger over it, you get I divided by M. So image divided by magnification. And then if you want to find magnification, it's image divided by actual. Image size, it should be, divided by actual size. Right, usually you get your answers in millimeters, okay? So you would use a ruler to measure in millimeters. So if you're measuring an image, usually they say convert some millimeters to something called micrometers. Okay, that's terrible. There you go, which is a micrometer. Can anyone remember the nifty trick you can do to convert millimeters into micrometers? Yeah?
You times it by 10 to the minus 3.
not to the -3. It's times, so if it's millimeters, you times it by 1000. Okay? Oh, unfortunately, just finish that. This is my wife. Sorry. Take it.
So do you have to put it into microscopes before you go around?
Right, sorry about that. Right, okay, so that's all I'm going to do for that. So, okay, so I'm just going to briefly just go through stem cells with you, okay? So we're going to do stem cells. Right, so can anyone remember what stem cells were? Yeah? They can, so they can become any other type of cell. Well done. So things basically, so they're what they are, undifferentiated cells. So they can become differentiated or specialized into any other type of cell. They do two things, okay, so they can either self-renew, okay, so they can self-renew, or they differentiate. Those are the two things they can do. Okay, so they can be used in medicine, let's say, so we'll just talk about medical use. So they can be used for research, okay, so you might use embryos, embryo stem cells. Where do they get embryo stem cells from? Can you remember? Yeah. Well, yeah. So they would get an embryo, though, usually before they're thrown out from IVF, in vitro fertilization clinics. Okay. Now, okay, what's a problem, do you think, with using embryos? Why have people got a problem? Yeah. Yeah, there are, because each embryo could be destroyed. So let's talk about issues. So they include ethical. So the embryo is destroyed, potential life could be destroyed. The other thing you probably can't remember but it's in the textbook. It is also a problem of risk of transferring viral infection. So you could transfer viral infections, which is not a good thing. Usually they give you lots of information on these when you do the exam question. Okay, also it's, you don't know the long-term side effects of using them. Can I say, anything related to drugs, all right, or vaccines or anything else, that's a good one to remember, long-term side effects. Okay, unknown side effects. But there's lots of uses. I mean, the most important use is to cure degenerative diseases, basically. So you can treat a wide range of diseases, to reduce suffering. You could grow replacement organs in the future. So you wouldn't have to wait for an organ donor. Okay, so that's embryonic stem cells. Now, that's embryo actually. It's all getting a bit mixed up there. So the other type is adult stem cells. So adult stem cells. Now, there is a big advantage of using adult stem cells. Anyone think of the obvious one first? You use an adult stem cell. Okay, so I'll give you, I'll go up to our friend over there and say, can I take your stem cells? What can you do? Sorry? You can say no. Yeah, good. If you're an embryo, you can't give consent. Whereas as an adult, you can give consent. So that's the first thing. So a big advantage is that people can give consent. They can say yes, they can say no. They can give their consent, an embryo can't. Okay, it's established as well, so it's being used for leukemia patients. So it's established, they know it works, and it's safe, relatively safe. Nothing's safe, but it's relatively safe. Okay. There is a disadvantage though. They can only, so disadvantage, can only differentiate into certain types of cell. Into a limited number of cells. Oh dear, can't spell. Limited number of cells. Okay. So what scientists have developed is what we call IPS, or induced pluripotent cells. So induced pluripotent cells or therapy. Pluripotent therapy. There we go. Does anyone know what they do with that? Can anyone remember? Probably not. I didn't teach it very well, to be honest. So all you need to know is they take an adult stem cell, okay, adult stem cell, and they make it behave like an embryo. So they place it, so they place nucleus into an egg cell, okay, and then they, through treatments, they then make it behave like an embryonic stem cell. So it behaves like an embryonic stem cell. So if they can get that working really well, then they won't need to use embryos in the future. They won't need to use embryonic stem cells because they'll just use induced pluripotent therapy. Okay? Right, okay, so we're going to talk about transport across membranes now. Right. Okay, you ready to go again? So about another 20 minutes. Okay, we'll do this quick. Okay, just right. I am going to give you a really top tip here. Transport across membranes. I would say in cell biology, okay, it's in cell biology, but you find this everywhere in the spec, okay, because when you talk about lungs and gas exchange, when you talk about the movement of water in and out of a root hair cell or through a plant, or when you talk about active transport. So, for example, active transport occurs lots of different parts of the body. So transport across membranes comes up all the time, okay? And there are three main ways that things transport across membranes. And the first one I'm going to talk about is diffusion, okay? So, diffusion. So what is it, first of all? So we need to say what it is. Okay, can anyone remember what it is? Yeah?
Is it the movement of gases and liquids apart from water?
Not water, okay. So things in solution possibly, but not water. I would say the spreading, the spreading of gases or particles. So the spread of gas or particles, usually gas, to be honest, let's say gas. So the gas could be dissolved, but yeah. From what we call a higher concentration to a lower concentration. Okay, so if I was to draw it, I'm going to draw a really bad diagram here. So if I've got lots of particles here, okay, over time, those particles will start to spread from where they're in a higher concentration to a lower concentration. So start to spread out. And eventually they reach an equilibrium where equal amounts again. So you're going from a high concentration, okay, high to low, and it's moving until you reach what we call an equilibrium. Equilibrium. Okay. So, e.g., where does it happen? Loads of examples. So, gas exchange in the lungs, so lung gas exchange. So nutrients in the small intestine, so more small intestine. So through the villi in the small intestine. So that's in humans. In plants, get diffusion of gases through the stomata. So CO2 and O2 through the stomata. Oh, no, we don't do stomata now, do we? It's organisation. That'll do. Do the next one. So next one I'm going to go is osmosis. Right, this one's a bit strange, this, okay? So what is it related to, first of all? Whenever you see the word osmosis, yeah, it's always water, yeah, so it's movement of water, okay? It's always water, all right? If any water's moving, it's osmosis. You right, Connor, or anything? All right, okay. So it is the movement of water, so movement of water from a dilute solution. We'll talk about what that is in a minute. To a more concentrated solution. Well, concentrated solution. But what does it have to move through? Do you remember? Does anyone remember? Yeah? No. What kind of part of the membrane? What do we call the membrane? Yeah. Yeah, so that's through a partially permeable membrane. Through a partially permeable membrane. Now I'm going to remind you, I'm not going to do loads of stuff on the required practical, but you did a required practical with potatoes. You might remember. Yeah? So you might remember the required practical. So we used potato chips and we placed them in different solutions of sugar. We looked at how their mass changed. If you don't remember that, please do visit DrBiology. Okay, all one word, and I've got all the required practicals there on my YouTube channel. I always bring it up, don't I? Yeah. My students hate me talking about my YouTube channel. Yes. 533. At the moment. Right, okay. Right, so I'm just going to do a quick diagram here. So here's a, well, don't know, cell could be. I'm going to draw a partially permeable membrane. Okay. Now, these are water molecules, but they're in a solution of glucose or sugar. So here's some glucose. Okay, so we've got the partially ppm, it's called ppm. Which side is dilute? Which side is dilute, more dilute? Yeah. Yeah, so this is a dilute solution here. So it's dilute. This is more concentrated. Now it's partially permeable. What does partially permeable mean? What do we mean by partially permeable? Yeah.
Does it mean like it only lets some things through?
Yeah. So in this case, it's only going to let water through. So water from a dilute solution to a more concentrated solution through the partially permeable membrane. Now, obviously, molecules are moving all over the place. They don't choose to go through the membrane. But as you know, I think it's in chemistry, do you talk about Brownian motion? So random motion of particles? Possibly not. I'm a bit behind the times on that. So yeah, so you'll get movement through the partially permeable membrane. Glucose, in this case, glucose cannot pass due to its size. It's too large. Okay. Good. Usually the example they give you in your exam is things like root hair cells. Okay, root hair cells, the movement of water. They also do it in stomata as well, so guard cells as well. Be aware of that. So guard cells, they might talk about the movement of water in and out of the guard cells. So if it moves in, the stomata open. If it moves out, they close. So that's stomata. Okay. And last one is active transport. Active transport. Okay, so active transport is similar to diffusion. However, the similarity stops really in terms of it's the opposite. So basically, it's the movement of particles from a low concentration to a higher concentration, and it's against the concentration gradient. So it's going against a concentration gradient. So it's going from low to high. And for that to occur, you need what? What do you need? Yeah. You need energy, you definitely need energy, and energy is being released, it's not made or produced, it's being released by respiration. So energy released by respiration. Okay, never say energy is made or produced. It's not. It's released. It's chemical energy released by respiration. These other two, these are what we call passive, that don't require any extra energy. So passive, osmosis is passive, fusion is passive. It's what we call when no further energy is required. Obviously, some energy is required, but not from respiration, just from movement, random movement. Okay, so literally it's the opposite. So you need energy. So it's against concentration gradient. Examples are the movement of glucose in your small intestine. Also in plants. So yeah, here's a top tip. Never forget about plants, by the way. They always talk about plants as well as humans. A lot of people don't like plants, but there's always questions on plants. I like plants. So plants, they absorb mineral ions that are in lower concentrations. So they're usually in lower concentrations in the soil. The drains. Right, we're kind of running out of time. One thing we haven't done today is cell cycle and mitosis. That's something that we should go through. I probably will go through next week. Gas exchange, I'm just going to very briefly just tell you about gas exchange. No, I'm not going to go through all the examples, but the three things you need to remember about gas exchange, okay, is when you talk about adaptations of things, e.g. like the lungs, for example. So the lungs is usually a good example. But for everything that is exchanging gases really well, there are three key features, okay? And you can remember, just remember it like this. So one, large surface area, okay? So how do the lungs have a large surface area? What have they got a lot of? Yeah. Alveoli, you've got millions of alveoli that are microscopic, that increase the surface area. They've got a thin membrane, always thin membranes. Okay, so capillaries, for example, where they're diffusing gases from the blood, from the lungs to the blood, and then carbon dioxide from the blood to the lungs, thin membranes. And third, they always have steep concentration gradients. So how does the lungs and the blood, how do you have a steep concentration gradient difference in terms of gases? What's constantly moving? There's two things moving, one in the lungs and one in the blood. So the blood is moving all the time, okay? And when you breathe in, you're bringing lots of oxygen in to make a high concentration. The blood, there's a low concentration. Okay, so blood is moving. It's usually to do with blood. Blood's circulated to maintain a concentration gradient. There you go. I can't even speak now. 5 period day. You've had 6 periods, so there you go. Okay, I'm going to wrap it up there, I think. That's pretty much everything. Well done. So we've pretty much done you've done a whole unit in an hour, basically. Okay. Okay, thank you for listening, everybody. I hope you found that useful. I will be going through some other revision of paper 1 topics. Just to remind you, all of this is on DrBiology, so please do subscribe soon. I'll see you soon.